Lesson 1M, ribonucleic acid, also known as RNA. DNA is a double-stranded molecule composed of one strand of polymerized nucleic acid, hydrogen bonded to a complementary strand. It then twists to make the famous twisted ladder form. DNA holds an organism's genetic code, the instructions for making all of the proteins required for the organism's normal function. DNA is very valuable to the organism and is very fragile, so it's stored safely in the nucleus. However, proteins are made in the cytoplasm by an organelle called ribosome, so a copy of the DNA instructions for protein is required, and this copy is called ribonucleic acid. Other than the obvious difference in its name and the fact that it's single-stranded, there are some other major differences. RNA and DNA are both long molecules of polymerized nucleic acids. Nucleic acids are monomers composed of a phosphate group, a pentose sugar, and a base. While DNA uses deoxyribose sugar, RNA uses ribose sugar with a hydroxyl group on carbon number 2. DNA nucleotides come in two varieties, terpyrimidines, consisting of cytosine and thymine, and the purines, adenine and guanine. RNA also has two pyrimidines and two purine bases, with one major difference. The pyrimidine thymine is replaced with the base uracil. These bases are polymerized just as DNA is, through a dehydration synthesis reaction of two nucleic acids. This creates a phosphodiester bond with the removal of water. The long molecule of RNA that's created does not hydrogen bond to a complementary strand. Unlike DNA, RNA is a single-stranded molecule. To recap, RNA is single-stranded, DNA is double-stranded. RNA is a copy of the DNA that will leave the nucleus so that ribosomes can use them to make protein. The sugar in RNA's nucleotide is a ribose sugar, while in DNA it's a deoxyribose sugar. And in the place of the base thymine, RNA uses uracil. The last molecule we'll speak on is adenosine triphosphate, also known as ATP. It's the energy currency of the cell. It acts like the rechargeable battery of the cell and is used to run the metabolic chemical reactions of the body. The molecule is created in mitochondria by using a single glucose molecule to create 38 ATP. As you can see, ATP is very similar to the nucleic acid adenine, with the major difference being a triple phosphate group rather than a single phosphate group. The three phosphates bound to one another contain energy. When these bonds are hydrolyzed, there is a release of that energy to help power the enzymatic reactions of the body. This will convert the ATP to ADP, adenosine diphosphate, while releasing energy. The ADP is then in turn recharged at the mitochondria back to ATP. This is a never-ending cycle of discharge and recharge of this molecule to fuel the body's metabolism.